Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's dive in. Ask most biologists, and they'll tell you there's like one way bacteria make CTP, cytidine triphosphate. You know, that vital building block for DNA, for RNA.
Right, standard textbook stuff for Streptococcus pneumonia, the pathogen we're looking at today.
But here's the kicker, the really counterintuitive part. This super common bug, it was hiding a second, totally secret CTP synthase enzyme. Fundamental stuff, just missed.
And they didn't find it by, just looking for enzymes. They found it by systematically breaking the cell's backup systems on a massive scale. That's how this secret gene, PyRJ, popped out.
It's an amazing story of how understanding gene teamwork, what scientists call genetic interaction, can literally rewrite biochemistry.
Absolutely. So that's our mission today, this deep dive into the method dual Tn-seq that mapped nearly all the genetic interactions in S. pneumonia.
Okay, before we get into how they did it, maybe just quickly define genetic interaction again.
Sure. So it's when you have two mutations, two broken genes, let's say, and the effect when they're together is totally unexpected compared to their individual effects.
Like the classic example, right? Synthetic lethality.
Exactly. Gene A knockout, cells fine. Gene B knockout, cells fine too. But knock out both A and B.
The cell just dies.
Right, and that tells you A and B were doing something similar, something redundant. You only see how essential that function is when both are gone.
And this redundancy is why so many bacterial genes are mysteries. Like up to a third of them.
Pretty much. We call it genetic dark matter. Knocking out one gene does nothing, so its function stays hidden. And traditional methods like transposon sequencing or Tn-seq, they're great for single genes.
But not for finding these double knockout pairs randomly scattered across the whole genome.
Exactly. Especially if the genes are far apart physically on the chromosome. It was just too hard to do systematically. We had maps like this for yeast, but bacteria, it was largely uncharted territory for this kind of all-to-all network map.
Until dual T-seq. So let's get into the method. This sounds like where the real leap happened.
It really is. The core idea was pretty ingenious. They combined standard barcode tagging, random barcode Tn-seq, with a genetic tool called the Krelok system. That combo made mapping these interactions massively scalable.
Okay, walk us through this. It sounds complex, but the goal is tracking millions of double mutants at once, right? How do they physically make them and then, crucially, read them?
Right. So step one, they create two huge libraries of S. pneumonia. In each library, cells have random transposon insertions, basically random gene breaks scattered across the genome.
Okay, random breaks.
And each break is tagged with a unique 20-letter DNA barcode. Think of it like a unique serial number for that specific break. Plus, these insertions carry special DNA sequences called lock sites.
So, library 1 and library 2. Millions of cells, each with one random, barcoded, lock site marked gene break.
You got it. Then they take advantage of something cool about S. pneumonia. It naturally grabs DNA from its environment. So they take DNA from library 1.
And feed it to the cells in library 2.
Exactly. The library 2 cells incorporate this DNA, and boom, you get this enormous pool of cells, maybe 1.4 billion, that now have two random barcoded insertions, two mutations per cell.
Okay, that's the massive scale. But here's the problem I see. Those two barcodes, those two insertions, could be like millions of DNA bases apart on the chromosome. How on earth do you read both in one go with sequencing? Sequencing reads are short.
Ah, yes. That is the absolute key trick, the bottleneck they had to solve. And this is where the Krelok system is genius. It's like a pair of molecular scissors controlled by a switch.
Okay.
They introduce an enzyme, Cre recombinase, which they can turn on using zinc. When they add zinc, Cre becomes active.
And what does Cree do?
It searches the entire genome for those two lock sites, remember? The ones linked to our barcodes, no matter how far apart they are.
It's okay, it finds them.
And it grabs them, brings them together physically, and performs a recombination event. It basically snips the DNA and stitches the two distant lock sites, and crucially, they're attached barcodes right next to each other.
Whoa, So it physically joins the information from two distant locations.
Exactly. You end up with a single short DNA molecule that contains both unique barcodes fused together.
And that can be read easily by standard deep sequencing. Very clever.
It's incredibly clever. It turns an impossible sequencing problem into a manageable one. And that's how they could sample almost 1 million gene pairs, 958,696 to be precise, covering about 73% of all possible pairs. It's a huge leap in coverage.
A category jump, as you called it. So they get millions of these fused barcode reads. How do they know if a particular pair represents a bad interaction, like synthetic lethality?
They measure fitness. They compare the number of times they actually see a specific fused barcode pair in their sequencing data.
The observed frequency.
Right. They compare that to what they expect to see based on how common the individual mutations were in the single mutant libraries.
So if a double mutant is really bad for the cell, it won't grow well, and its fused barcode will show up much less often than expected.
Precisely. If the observed count is way lower than expected, they use stats like read ratio and z-strains to check significance. It flags a strong negative interaction, maybe synthetic sickness, maybe full-on synthetic lethality. The combination is clearly hurting the cell.
Makes sense.
Yeah.
And did the system actually pick up known interactions? Did they validate it?
Oh, yeah, absolutely. That's the first thing you check.
Yeah.
And it worked beautifully. Known synthetic lethal pairs like PBP1A and PBP2A involved in making the cell wall, they showed up immediately as strong negative hits.
So the known weaknesses were flagged. Good sign.
And the data matched well with older, much slower one-to-all methods. It confirmed the approach was solid. Its sensitivity was around 65%, which is pretty decent considering the overall coverage was 73%.
Okay, proof of concept achieved. Then they looked at the whole landscape of interactions using COG categories grouping genes by function. What did that big picture view show?
It showed interactions everywhere: metabolism, DNA repair, building the cell envelope, you name it. But the most striking thing, the sheer number of hits involving genetic dark matter.
Those genes with unknown functions.
Right. The single largest group of interactions, they found 79 pairs involved at least one gene of completely unknown function.
And that's valuable because if an unknown gene interacts strongly with a known gene, it gives you a clue about what the unknown one might be doing. Guilt by association.
Exactly that. It's a powerful way to shed light on that dark matter. Like, they found 3 unknown genes, SPD 0751 to 0753, that interacted negatively, really strongly, with known transporters for glutamate, glutamine, and peptides.
Things the bacteria needs to scavenge from its environment.
Right. S. pneumonia can't make these itself. So the negative interaction suggests that when the main known transporters, GL, NPQ, AMI, are broken,
These unknown genes become essential, implying they're part of a backup transport system.
that's the strong hypothesis. Maybe an alternative peptide transporter or something related. It gives you a starting point, a function to test for these previously mysterious genes. It tells you how the cell stays resilient.
Okay, that's illuminating the dark matter. But let's get to the headline discovery. This secret CTP synthase, PIRJ.
Yeah, this one's pretty mind-blowing. They found this enormous negative interaction, like really strong, between PIRG
The standard textbook CTP synthase, the one everyone knew about.
and this totally uncharacterized gene, SPD 0300.
Wait, you're saying they missed a second enzyme making one of the most fundamental molecules for life, CTP, in this well-studied bacterium?
Because of redundancy. Knocking out PIRG alone. The cell managed, because SPD 032 is alone, probably also fine, but knock out both. Lethal.
Wow. And they confirmed it was involved in CTP?
Yep. The double knockout was lethal, but if they fed the cell cytidine, the downstream product, the cell survived. Clear proof it was involved in making or salvaging cytidine nucleotides. So they renamed SPD 032 to PIRJ.
Incredible. But there was a biochemical twist too, wasn't there? Something about how it works.
Yes, and this is crucial. They purified the PIRJ protein. Standard PIRG can use either glutamine or ammonia as the nitrogen source to make CTP, but this new one, PIRJ, it can only use ammonia.
A completely different chemical requirement for the same essential job.
A fundamentally different mechanism. It's not just a backup, it's an alternative pathway using a different substrate. It literally rewrites the biochemistry textbook for S. pneumonia.
And the implications go beyond just this one bacterium.
For sure. Homologs of PIRJ exist in other nasty pathogens like Clostridium difficile. So
Yeah.
if you're designing an antibiotic against CTP synthesis,
You can't just target the main PIRG pathway.
right, because the bug might just switch to using PIOJ and ammonia and your drug becomes useless. You likely need to hit both pathways to be effective. That's a huge insight for drug discovery.
Okay, so exposing a hidden essential pathway. Now, the second big find, YJBK and the cell wall.
Right. The cell wall is another key antibiotic target. Its synthesis starts with two redundant enzymes, mura and MRSA. In S. pneumonia, MRSA is usually the main player.
And dual Tn-seq found an interaction involving these.
It found that YJBK, another uncharacterized gene, had a strong negative interaction specifically with MERS, the dominant enzyme. But, interestingly, not with mura, the backup.
Okay, so breaking the main guy, MRSA, is okay because mura can compensate. But if you break MRSA A and D, this unknown YJBK,
Then it's catastrophic. When they did experiments to deplete MRSA in cells already lacking IJBK, the cells basically fell apart. They deformed and labeled.
suggesting YJBK is needed when MRSA is gone. But how?
Further tests strongly suggested something cool. YJBK seems to be required for the backup enzyme, mura, to function properly.
Hold on. So YJBK isn't doing the job itself. It's like regulating or enabling the backup enzyme mura.
That's the interpretation. It looks like YJBK acts as a kind of fail-safe regulator. It ensures the backup pathway, mura, kicks in effectively when the primary one, MRSA, fails.
So it's a gene dedicated to managing redundancy, keeping the cell robust.
Yeah.
Another layer of control nobody knew about.
Exactly. It reveals this hidden regulatory logic designed to handle internal failures.
These are fantastic biological finds. But zooming out, the method itself, dual Tn-seq, feels like the biggest story here. It's a platform innovation.
Absolutely. It's that category jump we mentioned. It takes us from looking at one gene, or maybe a few pairs, to mapping almost the entire interaction network of a bacterium in one shot. That's transformative.
And it's adaptable. Could be used in other microbes.
Yeah, as long as they're naturally competent or you have good transformation tools, the core Crelox barcode fusion trick is pretty universal.
So, thinking ahead, what does this unlock? What future research does it inspire?
Well, drug discovery is the obvious one. Identifying synthetic lethal pairs gives you ready-made targets for combination therapies, especially pairs involving these newly illuminated dark matter genes.
Hit two specific targets at once, and the pathogen dies. Much harder to develop resistance to that.
Exactly.
Mm.
And another big area is condition specificity. You can run this whole dual Tn-seq screen under different conditions.
Like low nutrients, or maybe with a low dose of an existing antibiotic.
Precisely. To see which genetic interactions become important only under stress or only when the cell is already fighting off a drug. That tells you about adaptation and resistance mechanisms.
It feels like we're finally getting the tools to draw the complete functional wiring diagrams for bacteria, understanding how all the parts work together.
It's a massive step in that direction, towards a truly systems-level understanding of how these microbes survive and thrive, or how we can effectively stop them.
So the final thought here, we've seen that even in a bug like S. pneumonia, supposedly well understood, there are these fundamental hidden backup systems, like an entire second CDP synthase using a different fuel source.
Yeah, hiding in plain sight revealed only when you systematically break the redundancy.
It proves that digging into these interaction networks doesn't just find weak spots. It uncovers the real hidden complexity of how life is wired. So the question to leave you with is, if S. pneumonia had secrets like this, what other fundamental alternative systems are still hiding undiscovered in the countless genomes we're only just beginning to explore?